So this is our third bit of trash within 2 hours on this dive, which is pretty upsetting.
Right, come on in.
As I said, spam.
What's the expiration date?
Sell-by date.
What is it?
No best by date.
Spam doesn't have a best by date. Yeah, that looks pretty
Keep
recent.
it. You could use that.
It looks like that was dropped in 1831.
Bring it up and have it for a snack tonight.
At least it's healthier. It was 25% less sodium.
I'm sure all of the animals down here really appreciate that.
Let's do a zoom on this guy. Let's do a zoom on this guy video.
Stand by for video.
Let's do a zoom on this guy video.
Stand by for video change.
Okay. Do a zoom on this, Tara, when you get an opportunity.
Absolutely.
Okay.
So that is what looks like holothurian poop.
All right.
So we don't need to focus in such detail on this. But thank you. Yeah, you can see the rest of it there in a little coil.
Oh, fish, center.
Got it.
Okay, so there's a fish just in the center of our screen.
Fish.
Right, come out a little bit, Tara. Yep.
Holding it.
So this is another ophidiid. Oh, they swim so beautifully and slowly. Everything is so much slower down here. And this is not the same ophidiid that we saw earlier. It is not Lucisaurus.
We are still in a serious move from the previous ship move.
Roger.
It's unlikely to also be Basozetus. So our onshore scientists are slightly puzzled as to what type of ophidiid this actually is.
2.
Okay.
Come out a little bit. Hold that.
Whoa, it's really translucent, isn't it?
Right, come on in.
You can see every little thing going on in that fish's body. Oh, that's amazing. That is some great imagery. Our scientists on shore should be able to further narrow down what fish that is in the future.
Come on out, video.
Oh, what?
All right, I'm looking straight down.
But the fact that they're puzzled makes me think that
60-meter
this is
move.
something perhaps not seen before in this area.
Yeah, I gotta push ahead. Snap zoom on this guy. Just to ID, and then I'm moving.
Sure.
So here's another holothurian. It looks pretty similar to the one we saw, the first or second holothurian that we saw, I think.
All right, that's good. Come on out. Gotta push ahead.
Thank you, pilots.
270, I believe. Correct enough?
Correct.
All right.
Looks like another one right up there.
So this area looks like we're sort of in a flatter area now, much more sediment coating everything.
Nav, bridge, move complete.
Thank you, Bridge.
a lot more fragmented rocks around. But they do look similar to the rocks we tried to collect earlier that essentially just crushed in our ROV's manipulators.
Yeah, if you see anything that looks good, we can stop and grab it.
And
I think just carry
just
on
a
for
note,
now. Deb's not actually here.
220 bearing.
What's that?
The waypoint 1 that originally we were trying to get is actually on 220 bearing, and we're looking at upslope is 26270.
We weren't able to understand you. Could you repeat that,
I bet if
please?
you ask watch lead if there's anything special there, just say we're trying to go uphill.
I'll talk to him.
Yeah.
Well, would you like a rock here? I mean, this looks a lot like the other stuff we tried to collect before.
They're coming out. Hold that.
Watch lead nav.
Sorry, Nav, what was that?
So I have a question for you. Our waypoint 1 is at a bearing of 220 and upslope is about 270, so we're not even, exactly heading straight forward. So how important is waypoint 1, or would you rather go up the slope and see what we're doing right now?
This is going to be a really silly question. Is waypoint 1 our end waypoint?
Let's get this guy in the water column.
I see it.
I
Nav,
believe it
is waypoint 1 our end waypoint?
Yeah, I'm double checking right now.
If it is, then we need to head towards that, because that will ensure that we see both soft sediment habitats and hard sediment hard
waypoint 1
substrate
seems to be our
habitats.
only.
Yeah, so let's head that way. Please, thank you very much.
Yeah, that looks like it's somewhat uphill also.
So this is a polychaete swimming
Oh, did they have backscatter
in
that we're
a
trying to cover?
rhythmic. Okay, my PhD supervisor, who is a polychaetologist, would not be happy with the fact that I cannot identify this polychaete.
Do you have any more zoom?
No, that's full.
That's it. Okay.
I'm thinking perhaps it's a type of acrocyrid.
Nav, can you zoom back in on HEPAC?
Sorry about that.
Coming out.
Yeah, I'm pretty sure that what you're looking at now, all sediment, just a real thin veneer of manganese, black manganese. But the blocks that we saw on the right-hand side previously are quite different. Those look like rocks. These look like sediment.
So for those of you that didn't catch that, I think Paddy was saying that here you can very clearly see, because of the sort of cross-section through the rock here, you can clearly see the manganese coating on the top layer of rock.
And also see black, black vertical cracks in that. And I think think what we were seeing previously in the surfaces of black prominent lines, probably was manganese
camera is at 90
fractures
degrees.
and sedimentary rock. And
We should start seeing
biologically
second
mediated
effects
by
about a minute or
microscope. It may just
Again,
rip
I
out
appreciate it.
individual cracks a little bit, so create these that we've been seeing.
So Nav, just to confirm, let's try and stay on course to get to waypoint 1, please.
Copy that. So pilots, I don't know if you heard the conversation we had earlier, but it is important for them to get to waypoint 1 because it'll allow them to see 2 different types of areas.
Quick zoom here.
And that's at 220, so we may think about changing our direction after.
Yeah.
Okay. Yeah, after this move, I'll
Yes.
move my heading over. Actually, if you look at my sonar, it looks uphill-ish.
Perfect.
But you said we still have about 30 meters left in this move?
Yes.
Okay, so I won't turn yet.
Let's do a quick zoom here. Partial. Yeah, hold that. Just making sure that's not fishing line. Alright, come on out. A lot of cracks in this.
So that, just going off the side, looks like another one of the jellies we saw earlier. We don't have to go, pilot, don't worry. So, can we look at what that black stuff is? Is it just that it doesn't look like a shadow? No. Is it? Okay, then don't worry, pilot.
Quick zoom here. Hey, hold that. Yeah, it's like a little sliver of
So right now we're looking at this this outcrop. Patty, do you have any thoughts on what might have formed this? I was out of the room, so if you already told them all, please feel free to ignore me.
Come in tight.
Oh, no, that's fine. Yeah, what I was saying earlier was that I think the vertical cracks in the sediment where microbes are more likely to be mediating deposits to manganese oxide. So this kind of looks like it's been a fracture or a remnant of a fracture filling. And you can see now, lower down in the image we're looking at, that there are many places where the sediment is fractured and there are prominent, more resistant ridges. And I think probably all that stuff is manganese.
Yeah,
Thank you.
Yeah.
I should also just mention, I think the fact that we see individual blocks of sediment in these last few images we've looked at here looks like this might be a mass racing block of landslides or undersea landslide blocks that's all broken up.
Sirius should be nearing the end of its move. About a minute.
All right. So the new direction is about 220, you said?
Yes.
Pilot, if you're comfortable, I can start slowly turning that way.
Have you stopped drifting?
I think I'm still going.
Yeah, he's coming up to the end of his move right now.
Okay.
It's about halfway angle change.
Do you have enough time to zoom on that sponge in the center?
ROV, can we look at that sponge in the center?
Where are we?
I think it's like center
center?
top right now.
I think it's a holothurian.
Do a snap on this and then
Sure.
just to verify it's a sponge. It is a sponge.
It is.
Oh.
Yeah. All right, come on out.
Sorry, that was our short zoom.
Yeah, we're coming in. We didn't believe you shoreside. We thought it was a holothurian.
That's a good spot.
Look at that geology in the background. This is very, very interesting.
All right, come on in, video.
We know.
There's something behind it.
So what's behind that sponge, shoreside science?
Full zoom.
Diva's coming back to tell us what it was.
This looks like a
Somebody typed
monopsid isopod. I'm thinking.
On the from the chat, it says a euplectidae.
Right, so
I might
so
be
that's the
pronouncing-
sponge.
Oh,
so
that's
the
the
sponge is
sponge.
a euplectellidae,
Okay.
and the isopod is likely a monopsid.
Come on out just a little bit.
Sorry, what?
Yeah, keep going.
Come on out just a little bit. Yeah, hold that. All right. If science is happy.
Scientists are happy.
Right.
So we're having a little bit of a controversy over here on the geology side about whether these little resistant ridges that you see that are sort of mesh-like, whether they are manganese coating or whether like a crust that's formed in those fractures, or if they're a silica. And we wouldn't know the answer unless we pick something up.
I bet you could break that one by your right skid with the skid, if that's what they wanted. If you just set down on it, whatever.
Are we in a position to keep moving, or are we waiting for something here?
I believe we're trying to break off a rock.
Oh, you are. Fantastic.
It's really soft.
If it crumbles, it's probably not something we want.
It's definitely crumbling.
So that's right there is where I just hit it with my skid. You want to come in, video? Yeah, come out just a little bit. Yeah. So that's what I just did there.
Yeah. I don't think that's what we want to collect.
I think I knocked that piece off right there.
Yeah.
I guess. All right.
So I'm facing 220.
We'll center up on you.
Okay.
Are we moving in that direction? Have we started?
No,
we have
haven't
not started yet.
Okay.
But I can call it in whenever you're ready.
Yeah.
Yeah,
we're
we're
ready.
Okay. Bridge nav.
Go ahead.
Can we get a 30-meter ship move at 220 degrees at 0.2 knots, please?
Ship move 30 meters, bearing 220, speed 0.2 knots.
Good copy.
Can you do the math and figure out how fast we have to go to make the final waypoint?
Sure.
Bridge move initiated.
So from the event log, we're hearing that a lot of these are probably submarine landslide blocks. There's a lot of seismicity in this area. There are many earthquakes. This is a tectonically controlled canyon, or fault side. So we might just be looking at things that have mass wasted down the side of the slope.
No. All right, I'm gonna come into this guy.
Okay. Do I set up?
Yeah, come on out. Come on out.
Oh. Couldn't hear you, pilot.
Yep, sorry.
Okay, pilots, we have to average a consistent speed of 0.15 knots. But that's
All right, come on in.
a solid 0.15 knots.
Mm-hmm.
Let me see what we've been doing.
So shoreside, what are we looking at right now? Diva's taking a well-deserved lunch break. What's an ACN?
Do you have any more?
So this is
No,
being
that's full.
identified as
Just very tiny.
an actinaria anemone, but not stalked.
All right, when you're happy, video.
We're happy.
A little.
We think it's an anemone,
Little
but we don't know what type of anemone it is.
It's been averaging 0.05
So this is an
knots.
anemone, but we're not really sure what kind of an anemone we're looking at.
Triple what we've been doing.
Don't think that'll happen. Is that a fish?
Apologies.
There's a fish bottom left.
Lower left.
There's another fish to the bottom left. Can we take a look at that?
Pilots, do we see a fish in the bottom left?
I see a fish.
We are on.
Yeah, come on in.
All right, so that's another Lucicorus. There seem to be quite a few of these in this location.
Okay, I'm gonna go That's full zoom.
All right, so we're
interesting. Mackenzie saying that Lucicorus, which I agree with. If you look though, this one doesn't have scales on the head, on the spout, and all. If I remember, there are two species in this genus, Luxiosus and Atlanticus, and I think that Jorgen Nielsen in Denmark, who described one of those species, said that Luxiosus has scales. This one doesn't, so this could be the other species, Atlanticus. We'll have to confirm. Atlanticus has been collected in the Solomon Sea in the South Pacific.
I'm okay with it.
Okay. Josh as well. Okay.
Well, just a little bit on the video. I'm losing my toe. Okay.
All right.
Okay, I'm centered on 220 and camera 70 degrees.
Do a quick zoom here. I'm going to pair whatever.
Bridge nav.
Go ahead.
Hold
At the end of this move, we'd like to put in another move of 30 meters at 220 degrees at 0.2 knots.
30 meters, bearing 220,
Can we do a quick
0.2
snap
knots.
zoom on that stalked animal, please?
Coming in on it.
Okay, so that is what looks like a sponge. A cladorizid, perhaps, with, I think, amphipods living on the stem. 3 amphipods. Yeah, it's definitely a cladorizid sponge, and they are actually the predatory sponges.
That's full zoom. Never seen anything quite like that.
Is it possible to get a zoom on the amphipods a little more, please, before we skirt off?
Yeah, Roger.
Want me to
Come
come
on out.
wide?
It's very small.
Alright, come on in video.
Full.
They want the amphipods.
All right.
Get them.
That's great, pilots. Thank you. One of our onshore scientists has just said that regardless of how good an image we get of these amphipods, it's unlikely that we'll be able to identify them from that image. And that is because a lot of the taxonomy to distinguish between different species depends on the mouth parts of these animals. But Les Watling is saying that these amphipods look like cynothoids. So thank you both, Mackenzie and Les, for that information.
Do a snap zoom here. Hold that. It's just rock. Okay, come on out.
So if you're just joining us, we are at 4,935 meters depth in the Marianas Trench, the south end of the Marianas Trench, at a site known as Sirena Canyon. During this dive, we anticipate heading upslope to hopefully see some hard bottom communities, which we are seeing now, and hopefully we'll also see some soft bottom communities, which should have a different fauna living on it. We've seen quite a bit of diversity so far, many different animals. Sponges, holothurians, crinoids, quite a bit of fish. And the geology has also been fascinating. So as we move up slope, we're going to continue to see hopefully some more animals. And I think we have an hour and a half left in this dive. Is that correct, Nav? An hour and a half?
Yes, hour and a half.
Yes, so an hour and a half left on the bottom here in this dive. And this dive may seem short, but that's because it's taken us a full 3.5 hours to descend the 5 kilometers down to the seafloor, and it will be more or less the same on the way up. We've collected 2 samples so far, a geology sample, which is basically a rock, and a volcanic nature, and also a crinoid, a comatulid crinoid. So there's a copepod just gone off screen.
Gosh, I don't know what that is in the bottom left of your screen, but it looks like it's alive.
Sorry, I lost the
Something white
shrimp.
and long in the lower left-hand corner. It could be another fish.
Another fish.
That's probably what it is.
I think it is another fish. And once we finish looking at this fish, pilots, we are happy to move on further towards waypoint 1, please.
Yeah, copy that. The ship is, we're continuing our move now.
Great.
Yeah, we have a 60-meter move in right now.
Great.
Come on in, video.
Zoom in.
So this looks like the same fish which we've seen several individuals of before. Definitely an ophidioid, probably another Lucicorus. And you can tell by the shape of the head,
Thank you, Bridge.
as I'm sure Bruce was saying earlier.
Come on in.
Yeah, that large eyes, which is
Hey,
unusual
co-pilot, are you seeing
for an
Sirius
ophidioid
moving
at this
already?
depth. I think it is Lucicorus. And again, we see that it doesn't look at their obvious head in the snout.
Yeah, I think I'm somewhere in that 60
Which means that
meters.
This is probably
I've been
a different
pulling up a
species than
bit.
the one that we saw on the island. One that's in the ocean and the Solomon Sea. The one that we saw on the islands is one that one from the eastern geographic sense, because they're, even though the Hawaiian Islands have largely an Indo-Pacific fauna, affinities toward the west, there are a few eastern Pacific species that get over in. We know so about the biogeography. It's really a deep living fish, and it's all speculation now.
Great. Thank you very much, Bruce.
I also want to just add a comment about the fish that had us cold. The other, what we thought was making a really interesting suggestion. She suggested that that could be Okeanos, and so that was the first observation of living fish in that family. Abyssal, generally an abyssal group of fishes. They've mostly been collected with nets that have been open, and it's always been a question of where in the water column they're collected, if they're collected near the bottom or if they're more of a pelagic species. So if that turns out to be an Okeanos that we saw earlier, that would be just an interesting hint from one sample about what the habits and habitat of that family actually is.
Come on out. Come on out.
Well, maybe by our next deep dive, you and Jeff and Mackenzie will have an updated ID for us. By our next deep dive, not just our next dive.
That's a new fish.
So this is definitely a fish we haven't seen yet. It doesn't have the eel-like tail. It has more of a conventional fish tail, and it's got some very bright white spots on it. Possibly parasites? Don't know. Let's see what our fish experts are weighing in as.
Coming down.
I'm wondering if this could be a thing called the Stanobaricid.
So pilots, we are done with our ship move.
Related to the
Say,
got it.
things that occur in shallow water.
That's full zoom.
Just 8 minutes or so left of Sirius
See what the
moving.
other fish people.
8 to 10 minutes. But in a couple minutes, if we're ahead, I can put in
I
another
see
ship.
the Jeff that this
So-
O'Barrett.
That's a good idea.
The Stanobaricid genera, and it's sort of from waters offshore. And then the western north is a very low possibility. There are some videos of fish in this family from a distance, so that's probably the best video that we have of a fish in this family.
Great.
Come on
Okay.
in.
Well, pilots, we are good to go ahead whenever you are.
Ah,
It's okay. I think we got some pretty great video of it so far.
last attempt. And
No, he doesn't want to be filmed today.
okay.
But that video makes me confident that the family identification is correct or extremely likely to be correct.
Cool.
So Nav, how much distance is it to get to waypoint 1? And what kind of speed do we need to be traveling at to get there?
We still have another 320 meters to get there.
And that should take us how long?
Should take about, like if we go, we need to average about 0.1 knot to get there before we head up.
So do we have time to stop for another sample or two whenever the ship is ready and you're ready? Oh, I guess it's one sample. You guys have both your bio samples, right?
What do you
No,
guys
we
think,
have one.
pilots?
Oh, okay.
Unless you collected
Kind
one
of-
when I was...
I think if we stop for another one, it'll be pretty rushed getting there, so
Okay.
probably not.
Yeah, I do think we should try and make some headway now to
Okay.
get up to the soft sediment community.
Yeah, but then we're going to lose our chance to get a rock sample.
Oh, yeah, of course.
That's so. If we once we head over that, we won't be able to probably pick up anything.
I bet we can work in a rock sample if we stretch out. And
Okay.
Yep.
And I'll go ahead and call on that ship move in just a second here.
Yeah, you can call it in.
Okay.
well, I guess ask if she wants one
Yeah, that's what I was
from here.
Survey, or watch lead.
Yes.
If you want one from here, we can actually spread out, get ahead a little bit, like right now. Try to get one.
I think let's try to get something.
Okay.
So geologists on the line, we were originally going to try to get one of those layered sediments or layered rocks. Is it okay if we try to grab something here? Anyone? If we could crack off like a piece of that that's coming off of our, like the lower left side.
This is sediment from this area.
Patty, can you type this into the event log? Because I can't hear you. So the UHECC, we're having a really bad connection with you. So if you could type it into the event log, that would be a better way probably to communicate right now.
Oh, Carl, what do you think? I don't see us breaking off that.
No,
Alex.
there's this.
You see
just
anything
set
loose
down,
at all?
It didn't break. So. But if you search around, do a little zigzag ahead of us. I'm starting to catch up with you. We can see if we can find one in the next few minutes.
I mean, there's that piece right there in the lower right that looks like it came off.
The one that's like just out of view right now?
Yeah, to the right. I mean, looks like it fell off.
We could try grabbing it and seeing if it remains intact. Oh. So Patty would like to keep moving north, further up the slope, because she thinks these are all sediments.
Roger.
Patty, I think Bob might have been interested in a sediment sample, but we can keep moving.
Thank you, pilots.
Okay.
Bridge nav.
Go ahead.
Can we get a 30-meter move at 220 degrees at 0.2 knots, please?
30 meters, bearing 220, speed 0.2.
Good copy.
Yeah, it sounds like they want one that's not crumbly, but also that we can break off, which I think is
Nav
impossible.
bridge, move initiated.
Or improbable.
Snap zoom?
Mm-hmm.
I think it's the holotherm we've already seen.
I think you're right.
Yeah.
So again, this is another holotherm, and you can clearly see he's had a lot to eat recently.
Right.
Biology, sometimes gross, but mostly interesting.
I mean, I feel like that's
soft up here.
not that bad. Yeah. Okay, we're approaching something that's stalked.
They come in partial video.
Zoom in partial.
Ooh, is that a benthiktinophore attached to a stalk? I think it is.
It's like a tunicate or something.
Yeah, I'm going to set down here briefly.
Yeah, definitely. Benthiktinophore.
That's cool.
Just don't get ensnared by those.
Yeah, I don't know how long they are.
How, can we have the laser scales on when you have a chance, please, pilots?
Absolutely.
I believe they're still on, yeah.
They come out real quick video.
Sure.
That's about the scale.
Okay, so it's pretty big, and
All right, you can come back in.
hello?
From Hawaii.
Hawaii, could you type it into
Beautiful,
event
beautiful
log,
pilots.
and we'll just read it?
Yeah, UHECC, your connection is terrible with us. We are getting the IRC pretty clear, but we just can't seem to understand anything that you guys are saying to us.
I think they're asking if this ctenophore is on a dead sponge stalk.
I think it is on a dead sponge stalk.
And then there's a question of what
And
the
they're
dark
also
purple
hydroids.
is. What is the dark purple inside the ctenophore?
I have no idea.
Okay.
Maybe some kind of nervous system.
Someone on the shore
I
side,
have no
anyone
idea.
want to comment on what the purple thing might be?
There's a long fishing line. Let's do another close-up.
So Mackenzie thinks it may be a platytonida, benthiktinophore. And you can see the strands it has hanging off it are what it uses to catch its food. So this is related to, you know, sea gooseberries and
what?
The other. Never heard of sea gooseberry?
No.
It's like a common name for a ctenophore.
Oh.
They're the ones that really shimmer and they can be lobed. Okay, don't know why I'm explaining this to geologists. I'll just give you a
The geologist is stumped over here.
But this is one which remains in situ.
All right, you're happy, science.
Yeah, that's great. Thank you.
Pretty.
All right, should we boogie for a little bit?
The
Guys, I can go ahead and put
thing
it
to
up.
look at on these guys are all of the teeny, projections coming off, and
So
there
we've
are
just
some of
never really
these
stopped.
that have them and some that do not,
far have we
and
gone in
which
this,
is,
at
I
220?
think the genus Hyroxes actually does have them, which it looked like this one had them too.
So if you
Okay, Chris, that
measure
was
from the ship to
really
that
clear
corner, what
compared
is
to
it?
previously.
Yeah.
So I think if you guys have reset your telecon,
No,
it is
I'm just looking
much
at the ship.
better.
Yeah.
And thank
To
you for
where?
that
To the
information.
corner of the
So we're just continuing
took
to move
a-
up slope a little
went
bit.
from
And
270
there's a
to
really
220.
large stalked animal that we're coming upon.
How about the really
87
large rock?
meters.
So we've done a continuous.
And it's on a
87
really
so
large
far.
rock. Or a piece of the
It's been, well, and since our,
trench.
so at 2 o'clock we put in a move. And then at 2:14 we put
I
in
think
another
this
move.
might be a fish.
Yeah.
To zoom down here.
Gotcha.
So I was saying in a couple minutes.
Patty, if we can find a sample here, would this be a good place to grab one?
Great spot,
With all this
pilots.
talk of sampling, because then we'll get to 120. It's basically continuous because you never let the ship
Exactly
stop
my
it.
thought.
Was that a yes?
I think we should do that.
Okay. So, pilots, if we have time here, it would be great if we could try to pick up a rock.
Pretty,
Roger.
Well,
far.
okay.
But only if there's time.
Nav bridge moving complete.
And a rock we can find that's willing to come with us.
We did stop for 15 minutes.
Oh.
We stopped for a full 20 minutes between ship moves. So we've had 30, 60, we've had 90 continuous.
Okay.
So this is another Ophidiad.
Yeah, that stop was at
And
the
it's,
corner, right?
So we
I mean,
stopped
out of
right
all
here.
the fish we've seen so far, which I think is about
really?
6,
For-
7 or so,
For
come
10
on out.
minutes.
all except for one have been Ophidiads, if I remember correctly.
So one of these
So
rocks,
Mackenzie's
maybe?
saying that she thinks that Ophidiad
If
densities
we
and
could get one, that would be
abundances
great.
in our deep sea are pretty underestimated worldwide. And it's the same one we were seeing before, Lucicorus, sorry.
All right, let's see if this this set of rocks wants to be taken away more than the last set. And then there's a request that we image the crinoid and whatever was next to it at the top of the block.
Yeah,
I mean, that's a
copy
pretty big crinoid if it is a crinoid.
I didn't call it a
No, I
crinoid.
know, I know.
You going
I'm
for the
just
one to the left of the
expressing
lasers?
that. I'm going to
skepticism.
try one of these three in here,
Okay.
if I can get set up.
So there's a little polychaete also swimming in the center, just by the lasers. Not important, just concentrate on the sample, I think. Probably related to one of the ones we've seen before.
Hydraulics on?
They are.
Can I get minis loose?
Go. Oh, yeah. You're gonna poke, then open the box, and then put it in?
Yep.
Okay. Let me know if you want me to tilt up or anything.
Okay, so we're just trying to figure out if we can grab one of these rock samples. It moves.
That one's loose.
That's a good news. Patty, what do you think? Do you like that rock?
That one all the way on the right is like
That one
the
moves too.
meatiest.
It's an abundance. Patty, I think you're going to have to write it into the event log. No, not that one.
Oh, you got it all muddy.
Nope. Okay, maybe not for this area. These guys are just too fragile.
Patty says if they
We can try going upslope a little bit more because these are just turning into sediment as we crush them. So thank you, pilots.
Okay. Sorry, watch lead.
Oh, not a problem. Thank you for trying.
So we have a sponge and we have...
Yeah, if we could just quickly zoom on that, but we're more interested in the humongous one up above. But they're probably the same now that we've seen this one lower down. Okay, so that's a sponge, a stalked
fish too over on the right.
A stalked sponge, maybe Hyalinema or Caulophacus.
Yeah, quick zoom on the head.
This is Caulophacus. I punched it in already into the event log.
Great, thank you, Chris. So there, yeah. So Caulophacus, we don't need to image the fish because it's the same one we were looking at before. So, oh, there's also a Brazingid. No, crinoid. One of the same is that a crinoid or a comatulid? I don't know. So, okay, so this is another Caulophacus sponge on an extremely long stalk that seems out of proportion to its disc. I've never seen one with such a long stalk before. And then I think next to it on the seafloor was likely one of the same crinoids that we've already collected. Maybe.
Okay, let's come down to the crinoid. We don't have much time here. The ship's moving serious. Okay.
Yep, one of the same crinoids which we already have in our bio box. Well, closely related if not the same. But it does look very similar. Yep.
So I'm looking
90, 80,
81.
So
and,
a crinoid,
I've
so
got
the
my
same one we
heading around
collected.
10 degrees.
So thank you very much, pilots. We can continue moving on.
That's good, because I have to boogie.
Great.
And get rid of some of this dust that's still on my arm. Let's do a zoom as I go by this guy. Stock crinoid.
Okay, so this is another type of crinoid. So this is the third crinoid I think we've seen during this dive. Third different type, and so this is a stalked crinoid. I can try to find out what a further classification.
It's something on the lower...
But if we need to keep moving, that's okay.
Yeah, as well. And it just looks like its stalk is overgrown with little hydroids. Maybe a Bathycrinus.
Okay, coming up.
So that is our third
The last
crinoid
shipment was 15 minutes
species observed during this dive.
Okay.
Sonar looks good.
So Alex Kerr, an echinoderm expert
much been
from,
90
I
degrees,
think, underwater Guam, please correct me if not correct,
time.
has said that
But we're shooting ahead. We could do another move, I think.
that stalk crinoid did not look like an isocrinus,
Bridge nav.
and therefore it is a new record
Can we get
for
a 30-meter ship
the Marianas
move at
region
220
for crinoids,
degrees
which is
at
great news.
0.29,
Thank you, Alex,
please.
for providing that.
30 meters, bearing 220, speed 0.2.
Good copy.
So correction, Alex is from the University of Guam Marine Lab, but is currently viewing this at Underwater World in Guam.
Do a snap zoom from here. Is that a stalk or is that a line?
So that to me looks biological.
Okay. Didn't want to get close to it if it was.
And then there's also another one of these crinoids next to it.
Something hanging on the stem.
Yeah, a couple cabetzals on the stem. What look like two amphipods further up close to the top, and something else a little further down, which is more difficult to see.
About as close as I can get right now.
That's great. No, that's great, pilot. Thank you. Whenever you're ready to move on, we are as well.
Yeah, we're just waiting for the ship.
Oh, sorry.
Yeah. Quick zoom in this guy. I think it's another swimmer. Yeah, another one of these guys.
So another one of these swimming polychaetes, likely from the family Tomopteridae or Acroceridae.
That is really cool.
But it's like a lava lamp, you know, you just want to stare at it.
Yeah, all day.
All day.
That's really cool.
Right.
Great imagery. Thank you, pilots.
Yeah.
An interesting comment from Les Watling at the University of Hawaii ECC. He says that whenever we see the pairs of amphipods, usually we see two amphipods at least on the stems of crinoids and sponges. Whenever we see that pair, it suggests that it's a male and female partnership. And that's been seen before with amphipod pairs on stalks of
There's a lot of loose rocks.
the type of sea pen umbellula.
Any of these rocks look interesting?
I'm not sure. Patty and Bob have any ideas on whether we want to try to pick something up here? This is really, there's a lot of rubble here.
This is also sediment.
You think it's all sediment?
Yeah, I think it's sediment, but if it's ash-rich, it might be of interest for composition. You know, just break it down and